Welcome to this biochemistry lecture on carbohydrate biosynthesis. There are two main ways which carbohydrates, in particular what we're talking about is monosaccharides, are produced. And honestly, we're really talking about glucose. And there's two main ways. There is the traditional way, which we've already talked about, which is gluconeogenesis. When I say traditional, I mean that's associated with mammals. And there's also a photosynthetic pathway that we're going to introduce to you, although as pharmacists, you are likely not to see this a great deal, obviously. So carbohydrate biosynthesis, basically we are going to take water and CO2, carbon dioxide, and we're going to provide energy in the form of sunlight. This is obviously the photosynthetic pathway, and we're going to get glucose out of it. We'll discuss gluconeogenesis, which is the other way, here in a little bit. In fact, right now. So as you might recall, we take pyruvate and we do the reverse of glycolysis, and we can get to glucose. And so we're basically going to take pyruvate, which is a three-carbon structure, and we can go backwards using many of the same enzymes, same of the main pathways, and we can get to glucose from pyruvate. I don't think I'm going to spend too much more time. I would know the enzymes for this reverse reaction for exams. The other main way is photosynthesis, and there's two types of photosynthetic reactions. One is a light reaction, where we can convert solar energy, which is sunlight, to chemical energy, both ATP and NADPH. And there's also dark reactions, also known as the Calvin cycle or Calvin and Benson cycle. These are light independent reactions, but they use the energy produced and stored from the light reactions to generate and incorporate CO2 into an organic molecule called pGal, which is later converted into sugar. That's the general gist. So there are two ways we do this. Again, light is going to convert into ATP and NADPH, and from there we take the Calvin cycle and we can generate and spin off all our sugars, and we can also spin off and we spin off and take those reactants and put them back in so they can be recycled so we can generate more sugar. So we're taking CO2, putting it in a Calvin cycle, and we get out sugar. Okay, so the first process in photosynthesis is the light reactions. The light reactions are called non-cyclic photophosphorylation. Photo because you're going to take light, phosphorylation because we're going to make ATP out of it. And so electrons are removed from water and they're passed through this system in order to generate that energy. And basically the steps here, so light's absorbed in this PS2 system. They're passed from pigment to pigment until they reach a molecule called P680, and from that high energy electron is passed to an acceptor molecule and replaced with an electron from water. So we split water and we get O2 in the end. The high energy electron then travels down the electron transport chain. It does lose energy as it goes. Some of the release energy drives the pumping of protons, and it builds a gradient. And again, in step 3, we're going to have another exception of a proton. Ferredoxin is then transferred. And from there, we're going to have a pump that's going to, again, generate ATP, just like the FOF1 ATPase does, or I guess I should say, in a very similar way to the FOF1 ATPase, but we're basically using electrons to be transported off in order to create a pump and to create NADPH and that proton gradient. The next is the dark cycle. So dark cycles, also called the Calvin cycle, it uses energy from the previous step to create CO2 and pGal. What you basically have, again, CO2 is then converted into glucose. So an enzyme that's abbreviated Rubisco, and ribulose bisphosphate, which is labeled RUBP, have 5 atoms of carbon and a phosphate group on each end. So Rubisco catalyzes a reaction between carbon dioxide and ribulose bisphosphate, which then forms a 6-carbon compound that is immediately converted into 2 3-carbon compounds. And this process is called carbon fixation because the CO2 is fixed from an inorganic CO2 molecule into an organic molecule. And ATP and NADPH use their stored energy, and that's how they're converting those the 3-carbon compound 3-PGA into another 3-carbon compound called G3P. And this type of reaction is a reduction reaction because there's a gain of electrons. So a reduction in the gain of an electron by an atom or molecule is the story there. The molecules of ADP and NADP are result, and they are returned to the light dependent. So they're returned to the light reactions to be re-energized. One of the G3P molecules leaves the Calvin cycle to contribute to the carbohydrate molecule. So that's commonly glucose, and so that's where right after the second stage, that's where that happens. So the carbohydrate molecule has 6 carbon atoms, and it takes 6 turns of the carbon cycle to make 1 carbohydrate molecule. So 1 for each carbon dioxide molecule fixed, and the remaining G3P molecules regenerate RUBP, which enables the system to prepare for a carbon fixation step. So in summary, it takes 6 turns of the Calvin cycle to fix 6 carbon atoms of CO2. And these 6 turns require energy from 12 ATP molecules and 12 NADPH molecules. That's the cycle in a nutshell. 3 steps. Step 1, carbon fixation. Step 2, reduction of 3-PGA. Stage 3, step 3 is regeneration of the ribulose bisphosphate. Other carbohydrates can be produced as well. Glucose can be converted to hexose and to di and oligo and polysaccharides. The common step in all of these synthesis is activation of glucose by uridine triphosphate or UTP to form a uridine diphosphate glucose, and from there enzymes can convert that into different structure 6-carbon carbohydrates. That's the end of this lecture. It's mainly an introduction to this concept of photosynthesis. I don't know if we'll spend too much more time on photosynthesis, but that's the story. I look forward